So it's really a pleasure to come back to Prague and to have the opportunity to visit you and your institute. So that's what I will talk about today, how we can study interactions between life and minerals down to the nanoscale using this kind of microscopes, so electron microscopy and more particularly transmission electron microscopy, and combine this with X-ray microscopy. And for the production of X-rays, we use synchrotron radiation. So please feel free to stop me at any time if there's anything that is not clear enough that I forgot to mention. So as a very general introduction, the focus I have had over the last few years is really the formation of minerals by microbes. So first message is that bacteria or microbes more generally, they can form minerals. So that's the first thing. They can form a large diversity of minerals, and this has potentially big impacts, several ones. And it is of interest to several scientific communities. So the first one, like this is a TEM, Transmission Electron Microscopy image of a bacteria cell that got encrusted in calcium phosphate minerals. And this has been observed in some pathological calcification in like arteries or things like that. And so some of health problems involving calcification might involve might involve some microbes. So that might be understand which microbes do that, how they do that, how can we get rid of these things. I mean, that might be one interest for medical science. This is another example. So this is a mine, the Rio Tinto mine, and you might know this, the site and the environmental problems, issues associated with this mine. There is generation of acid mine drainages. So you have those really nice red waters. It's really beautiful, and in the south of Spain, it's pretty warm. So usually you want to swim or bath into these rivers, except that the pH is pretty low. It's pH 1 or pH 2, and there is a huge load of heavy metals. So probably it's not a good idea to go there. But there are also many microbes in those rivers, and they are also and interestingly, in those rivers you have formation of sediments of minerals, and part of this process, part of the formation of the sediments involves microbes. And the result of this is that these minerals trap some of the heavy metal pollutants. So here again, the formation of minerals by microbes might be a way to bioremediate, but there are also some microbes that might be involved in the release of some pollutants in the rivers. So really, those microbes and the formation of the minerals or the weathering of those minerals might be involved in these processes. This is one other picture. These are bonded iron formation. So there's really huge sedimentary deposits, very rich in iron, that can be exploited, and so that are used as mineral resources. And there's still a big debate on how these sedimentary deposits formed in the past. They are several billion years old deposits, and some of the models propose or suggest that some microbes might be involved in the oxidation of iron that led to the formation of those deposits. So again, some microbes and the formation of minerals by microbes might be involved into the formation of this kind of deposits. You might know these objects, stromatolites, and you might know that stromatolites, we found some stromatolites in modern environments. So this is a picture in Shark Bay in Australia of some living stromatolites. And hundreds of kilometers away from that, we find fossil stromatolites that are 3.5 billion years old. And those stromatolites, they are suggested to be among the oldest traces of life on Earth. And the idea is that these are rocks, these are just big aggregates of minerals, and they might have been formed by microbes. So again, the minerals that are formed by microbes might be interesting if you want to look for or search for traces of ancient life. And it has also been proposed that if you want to look for extraterrestrial life, you might find this in rocks. And finally, some of the microbes. So this is again a transmission electron microscopy picture of a bacterium. So you see a cell here, and you see this here, these these minerals here that are magnetite nanocrystals. So those bacteria are able to form intracellular magnetite crystals. And the story is that they use these very small magnetite crystals as magnets to orient themselves in the environment. So basically they can feel the magnetic field and move along the magnetic field, and so find their way to the appropriate conditions for their living. And the interesting thing is that, of course, this kind of magnets we are very useful in some technologies. And so for years and years, people have tried to synthesize such crystals with very specific sizes, with very specific chemistry. And humans managed to get some protocols to produce some nice crystals. But those bacteria, they produce much better crystals. And so the idea is that if we understand how they manage to do these crystals, how they manage to control the shape and the size of these crystals, maybe we'll be able to also manage to do this the same way, and then to produce some interesting materials for new technologies. So here, this is really the summary of why should we care about formation of minerals by microbes. And this is what I care of, right? So it's not just, as you see, there are different fields of interest. And the interest is really to understand how microbes manage to produce minerals. So if we want to understand these things, if we want to understand biomineralization, how this works, there are several implications. I mean, we have several needs. First one is, here are some biominerals that you know of. So this is a scanning electron microscopy of diatoms, coccolith, or forams. We find them, they are abundant in the ocean or in lakes, and they form those very, very nice pictures, very nice structures. This is lovely. I mean, even if you don't know what is behind it, it's aesthetically interesting. And that's the first thing. How do these organisms manage to control the shape of these crystals? How do they do that? What are the mechanisms involved in this control? So that's the first question we ask. What are the mechanisms involved in the synthesis of these minerals by life? Second need we have is the following. So this is a list showing some carbonate minerals that can be produced by life. And what you can see is that there are different types of carbonate minerals that can be formed by organisms, like calcite, aragonite, magnesian, calcite, dolomite, strontianite, witherite, vatrite, and so on and so on. So it's almost the same, chemical composition. You have different polymorphs, but different structures. Some are well crystallized, some are amorphous, some have a different chemical composition. So if we want to understand the formation of minerals by organisms, we need to be able to study the chemical composition and the structures of these minerals in the system. So we need to be able to do that, discriminate between different minerals formed by these organisms. One nice example is provided by this trilobite. This trilobite, so they are fossils, of course. Sometimes the eyes are preserved, and that's how they look like by scanning electron microscopy. And you see these, so these eyes are composed of different units. Each single unit here is a lens, is a single eye. And these single lenses are calcite crystals. So now if you consider that this one single eye is a calcite crystal, you might know this very simple experiment that we use in classes. We take this single crystal of calcite, put it on the text, and ask the students to read the text. And of course, it's pretty difficult. This is, of course, one property of these kind of crystals. So let's imagine now you are a trilobite, and you see this way. That's bad. So it looks like when you come back home drunk or something like that. So it's really bad. So there's a way to use a calcite single crystal and to see things clearly. And for that, you need to orient this calcite single crystal in a very specific axis. If you look this way under this axis, it's okay. So now that's what they are doing. Those calcite single crystals, they are oriented in a very specific way so that you don't have this effect. So each crystal here is oriented along the ternary axis of calcite. So they are able to control the crystallographic orientation of the crystals. So now if we want to look at these things, again, we need something. We need to be able to characterize the crystallographical features and the orientation of the crystals in the systems. Finally, these are the kinds of crystals we are interested in. So again, these magnetotactic bacteria, these bacteria that form intracellular magnetites. The crystals here, so this is TEM, this is a TEM picture. The crystals here, sorry for the scale, but this is less than a micron wide. And those crystals here are a maximum of 100 nanometers. So if we want to look at those things, we need to look small. We need to be able to look very small. The second thing is that all those crystals, so this is nacre. Nacre, you see all these, this is SEM. You see the tablets here are aragonite crystals, and the tablets are separated from each other by organic molecules. And in all these systems, crystals are very intimately associated with organic molecules. So we need to look small, and we need to be able also to characterize the organics at the nanoscale. So this is really why we need to use microscopy. We need to characterize minerals, the orientation, the structure of minerals, the chemical composition of minerals, and the organics at the nanometer scale. So that's why we need to push microscopy techniques if we want to improve our knowledge of how microbes form minerals, how it works. I just make a very short note. Actually, it's not only of interest to people working on interactions between life and minerals. It's also of interest to people working on fossils. And okay, those are two fossils, but basically the questions are the same. If you want to better understand what is preserved in those fossils and how it got preserved in such a nice way. Here you preserve the bones, but you also preserved feathers and soft tissues. If you want to understand how this is possible, you also need to characterize the organic and mineral assemblages and down to the nanometer scale. So again, you need to use these microscopy tools to get into the processes that formed these fossils. Okay, so I think these methodological, technological improvements, microscopy improvements, have really marked the recent developments of that field. I'm really convinced of that. 1987, I don't know if you know this picture. So this is light microscopy, and this shows those filaments in 3.5 billion years old rocks, sherds. And those filaments have been interpreted as the oldest microfossils known on Earth. So they are, okay, light microscopy. And that was, at that time, that was one of that was one of the most important techniques that was used to characterize those things. Now, we have so many techniques that allow you to look at those samples down to the nanometer scale, analyze the chemical composition of those objects, analyze the isotopic composition of these objects, analyze the structures of the minerals down to the atom scale. We have so many techniques that are available, but that we need to apply to this kind of complex natural samples. So that's and if you do that, of course, you have a completely different view of what this is. So this basically should change our interpretation of all the geological records. Because it's like if you need glasses, just take them off and look at the world, how it looks like, and then put them back, and okay, you have a different view of the world, probably. Okay, so that's basically what I will present you today. Some examples, some illustration of how we can use these tools in order to better understand biomineral assemblages. So I'll show you that with very few illustrations, and you're welcome if you want to know more about each case, you're welcome to ask. So this will be the two main techniques I will talk about. So transmission electron microscopy, and probably, this one is probably, is more known by the community. And that one, this is synchrotron-based X-ray microscopy, or STXM. So for scanning transmission X-ray microscopy, the microscope is here. It is much smaller than this one, but very powerful. And I'll show you how we can combine both of them in order to characterize the crystallography of mineral phases, well crystallized or poorly crystalline phases. How we can use these tools to characterize the organic molecules, to characterize redox state of iron or other elements, and of course everything down to the nanometer scale. I won't dwell on this, but of course, those two microscopes I've shown you are transmission microscopes. So you need light going through the sample. You need electrons or X-rays going through the samples. And that's maybe an issue, a drawback with these techniques. For this, you need very thin samples. And when I say very thin, it's down to 100 or 200 nanometers. So this is really thin. So now you want to look at your samples, but you want to get very thin pieces, cuts in your samples. But we have techniques that allow you to prepare such samples, such cross-section. And one of them is quite powerful. It's the focused ion beam milling. So that's what I call FIB, for focused ion beam. So this is how it looks like. This microscope is basically a scanning electron microscope. Okay, so you can recognize, just remove that part, and this is a scanning electron microscope. So you use electrons to image the sample, the surface of a sample, very easily. Any sample, you don't need sample preparation for that. You just bring a piece of rock into the SEM and look at it. Now, in addition to the SEM, you have this, and this is an ion gun. So now in this microscope, you can also use an ion gun, so bombard your sample with ions. And if they are energetic enough, you can dig into the sample. You can dig into the sample, so you can look by SEM within the sample, and you can also excavate some cross-sections, some thin cross-section from your sample. So better than long discussion or these speeches, I'd like to show you that movie that illustrates how this works, how the sample preparation works. This was this movie was done by Hugues Leroux from University of Lille, and what they prepared here is a stardust sample. So they got some dust of the comets from space back to Earth, and the dust, I mean the fragments of the comet dust are they were collected on the gel. So the dust impacted the gel, and this is the impact crater of the fragments. And so what they want to do here is there is a fragment, a mineral fragment within the crater. They want to cut a cross section across this. So take the cross section and put it in a TEM. Okay, so this is in a FIB. So you see here, this is a scanning electron microscopy image. And here is how they do it. So, okay, this is just SEM picture. They demagnify. Now they rotate the sample and they go to the sample. First step, they will deposit some platinum on top of the sample to protect it. So they will deposit platinum in the FIB, and that's what you see. There is something growing. Okay, this is the platinum. Okay, there is a platinum trap that is deposited on top of the sample. Now they dig into the sample on both sides of the platinum strap. They use ions to dig into the sample. That's how they do it. So, okay. Now, what they are going to do is they will cut with ions. They will cut at the bases of the sample, of the section, and they will cut on the side. So that's where they are doing. Up, they cut here, they cut here, they cut on the bottom, and they cut part of it, but there is still a tiny piece that, help to keep the cross-section attached to the sample. Okay, so they do it. Now, okay, you see the sample. So now, demagnify, what they will do is that they will insert a needle, very thin needle, micromanipulator, close to the sample. Hop, here it is. So close to the sample, you see the tungsten needle here. And they will approach it, to the cross-section. And now they will glue the needle to the sample by depositing platinum. So that's what they are doing here. Okay, now they cut, and now they remove the cross-section. Hop. They bring it to a sample holder. This is for TEM. This is a sample holder that you can hold with your tweezers. And so what they do is that they will glue it. So they will glue the cross-section. So that's what you see. They deposit platinum here, and they glue it. And now they cut the needle from the section, and now they thin down the sample. Thin down to 100 nanometer. Okay, they do this. Once it's ready, you can get the sample holder out and put it in a TEM, and now you do TEM on the sample. And you see the cross-section. Okay, so now it's back. So that's how you prepare a sample, for example. The good thing is this: if you can see your sample by SEM, you can cut across it and prepare it for this transmission microscope. Okay? So your sample can be small, 1 micron. It's okay, you can prepare it. You can have one single particle in the whole sample. If you can see it by SEM, you can prepare it. And so this technique, you can use it not only for meteorite sample, you can also use it for interfaces between microbes and minerals, and you can preserve the contacts between microbes and minerals. And this is one example. Here, this is a cell, a fungus that is lying at the surface of a mineral, and you can cut across it, and this is TEM picture of this cross-section. And here you see the fungus cell, it's here. You see the crystal on which it is lying, and you see some crystals that are formed at the surface of these crystals and close by to this cell. So you can really preserve also those assemblages. Okay? Okay. So to illustrate then the use of synchrotron-based microscopy and TEM, I'd like to go through this first example, the study of fossils from metamorphic rocks. The general idea is the following. If you consider fossilization, this is the first step. You have the living organism. It dies eventually, and it gets trapped into sediments. That's really the fossilization process. Of course, this step is very unusual. You need very specific conditions so that it can work. But you can find these things in sedimentary rocks, and that's where paleontologists usually look for fossils, sedimentary rocks. Now, if these rocks go to deeper depths, it might experience increase of temperature, increase of pressure, some shear, some fluids, and so you get a metamorphic rocks, and all the information that is stored here about the primary organism will be blurred. That's, basically here you don't see much anymore, but there's still something. Our goal was to understand what is left in these rocks about this. Do we still have some information about this in this kind of rock? Because if we can get some information about this extends significantly the record for paleontology. I'll skip this. Basically, the idea is that metamorphism, when you increase temperature and pressure, there are many processes that can transform these fossils. You can transform the organic carbon, like going through these kind of molecules to graphite. So you really remove a lot of information. And metamorphism also can produce chemical composition that are similar to those formed by life. There are some reactions that can be triggered by metamorphism that produce organic carbon. For example, if you consider a rock that contains siderite, so iron carbonate, if you heat that up, you might produce magnetite plus graphite. So you might end up with organic carbon, but this is purely abiotic, not produced by life. So really, metamorphism can trick you and can really change the information you store into these rocks. So we looked at some rocks that we collected from the French Alps. This is how these rocks look like. These are carbonate concretions. They come from that level in the Vanoise. And what we know from mineralogy is that those rocks, they formed in the Trias, so they formed 230 million years ago. And those rocks, they went into subduction. They went down to 40 kilometers beneath the surface, and they experienced these kind of conditions, 360 degrees and 14 kilobars. If you look at thin section by light microscopy, if you look at thin section of these rocks, this is what you look like, what you can see. So you have a carbonate matrix, all the white areas, and you have many dark objects. Those are fossils. This is how they look like. You have this kind of objects, dark round-shaped spheres, and this kind of objects. Now, this is what paleontologists do, and they compare this to this. These are modern spores of some like fern-like organism, and these are tissues of also some similar plants. So the interpretation is that these things and these things that we find in the metamorphic rocks are remnants of this kind of objects. Okay, so they are fossils. Not very big, 150 microns, 50 microns. So not really big, but that's what we studied. You can start at a low magnification. You can use a Raman microspectroscopy to characterize the carbon that is composing these fossils. And this is the typical spectrum you get everywhere on the carbon that is in these fossils. And we can use this spectrum to assess the temperature that was experienced by these objects. It looks like, you know, this kind of spectrum changes with the temperature. So if you have low temperature, you have this kind of spectrum that looks similar to that one. And if you have high temperature, you end up with only this single peak, and this one is decreasing. So from this spectrum, so you can really use this spectrum as a geothermometer. From this spectrum, we could see that the maximum temperature experienced by these objects was 360 degrees. So that means that these objects, they are not just contaminants that were trapped into the rocks after the metamorphism, they really experienced the metamorphism. Okay, you can do mineralogy with the Raman spectroscopy, and this is nice. So you see that those fossils, for example, they are composed of an organic carbon wall, and there are also many different phases. There are calcite fillings, and there are also another type of carbonate, which is ankerite. This is an iron, magnesium, and calcium carbonate, which always shows up beneath the organic wall of these fossils. So you see that it forms some kind of corona here beneath the organic wall. Okay. Now let's go to FIB. We prepare a cross-section here across the cell wall of these fossils. So we dig into the sample, we get a cross-section, and this is Transmission Electron Microscopy picture of this cross-section. Here you have the inner part of the fossils, so which is filled by calcite and the ankerite, so the calcium, iron, and magnesium precipitate beneath the cell wall, the organic carbon cell wall. So TEM is very convenient to give you very nice images, nice resolution images, and do some basic mineralogy. This is a cross-section from the other fossils of the vascular tissues. You can do EDX mapping and see where the phases are. In that case, the mineralogy is different from the one I showed you previously. You have pyrite, iron sulfur. In blue, you have the organic carbon. And in red, something that we could not see by Raman spectroscopy, at the interface between the carbon and the sulfur, you have iron oxide. So you can do electron diffraction, you can do high-resolution imaging, EDX, and with that, you have a complete exhaustive description of the minerals that are associated with these fossils. Now, let's use the Scanning Transmission X-ray Microscope. So that's synchrotron-based microscope. This is how it works. So this is transmission microscope. There is an objective lens, so that focuses the beam that is coming from the synchrotron. It focuses the beam down to a spot of, let's say, a 25 nanometer. So it focuses the beam onto the sample, down to a spot of 25 nanometer. You have the sample here, and you look at the photons that go through the sample. Now, with this, you only have the image of one spot. You see one spot, 25 nanometer spot. If you want to get an image, you need to scan the sample in X and Y and record the photons that go through each pixels. So this is why we call it scanning, because you have to scan the sample, and transmission, because you record the photons going through the sample. So scanning transmission X-ray microscopy. This is done with a single energy. But now you can take an image at a single energy, and now you can change the energy and take another image at another energy. And so for each pixel of your image, you can measure the absorption by your sample of the photons versus the energy of the photons. So you get images with a spatial resolution of 20, 25 nanometer. And you get spectra. And these spectra, they are called Xen spectra for X-ray absorption near-edge structure spectra. And here what you look at is the absorption of X-rays against the energy. And this is the kind of spectra you get. And you see there are peaks, and basically these peaks tell you something about the chemistry, the electronic environment of the atoms. So that's what is explained here. You measure the number of photons that are absorbed versus the energy, you get peaks. And each peak relates actually to electron transition between orbitals. And that tells you something about the type of bondings that those atoms share with other atoms, with other neighboring atoms. So this will tell you something about the chemistry of these atoms. So now if we do this analysis, so we use exactly the same FIB cross-section as in the TEM. Now look at that. We measure the Xen spectra at these 3 different spots. They are very close by. They are less than 1 or 2 microns away from each other. If we look at the blue point, TM tells us that this is carbonate. This is how it looks like. We have this spectrum, and this spectrum is characteristic of carbonate. We know that this peak is characteristic of electron transition in carbonate groups. So, no news. Now, if we take the green and the red, we have those. So, red and green, we have these 2 spectra. Even if you don't know anything about Xens, you see that they look different. Here, for example, you have a peak. There is no peak here, and so on. So, there are some differences. Those differences means that the 2 carbon that we have here, the first thing is that those 2 points, they are taken on organic carbon. These spectra are characteristic of organic carbon. And these are 2 different types of organic carbon. They are different. So our interpretation was the following. If now you compare the red one with this one, they look similar. You find this peak, find a piece of this peak, you find this peak here. And this spectrum is a reference spectrum of sporopollenin. Sporopollenin is an organic polymer that composes the walls of pollen grains. Our idea is that initially this was some kind of pollen grain. The cell wall was composed of sporopollenin. Then it got transformed by pressure and temperature. So we transformed some of these functional groups, but eventually we find some remnants of these functional groups in the metamorphic samples. That means that sporopollenin is a very, very resistant molecule. It's one of the most resisting organic polymer that we know. And here it experienced 360 degrees, 14 kilobars, and it's still preserved. Some of the functional groups are preserved. In the meanwhile, there are some other areas where there is nothing anymore preserved. Those functional groups are not preserved anymore because this was, and our interpretation is that initially these polymers here were different. They were not sporopollenin. They are less resistant, and so they transformed more significantly to graphite. Not graphite, but they went to graphite. So that's the take-home message. If you look with the appropriate techniques, you can see that there are some chemical heterogeneities at the nanometer scale. Some of these heterogeneities, they concern organic carbon. Here, we find some chemical signature of sporopollenin, which is a very resistant polymer in these samples. And in the rest, we have more graphitic carbon. If you remember, by Raman spectroscopy, we only saw the structure of the carbon, of the aromatic rings, and we saw that basically it was homogeneous everywhere. If we've combined Raman with this technique, we see that there are some heterogeneities that we were not able to see with Raman. Okay, so that's the model. That was the initial structure of the spore wall as we know it from modern analogs. There was the sporopollenin forming all that thing. There was some cellulose here. Cellulose got degraded and replaced by ankerite. And the sporopollenin was transformed into metamorphic sporopollenin, so it's no longer exactly sporopollenin, but there are still some signature, spectroscopic signature of that polymer, plus some graphitic carbon. I skip these things. That helps. Can I take 5 more minutes?
Yes, sir. You may have 10.
10. Okay, so that was for the fossils. With all these analyses, basically that's what I didn't show you, but we can tell a whole story of how it transformed upon time. So we know that cellulose got degraded, replaced by some carbonates. Then it went to deep depths, 40 kilometers. Then there was some transformation of the sporopollenin into partly, and the other polymers into graphitic carbon. And we end up with something that looked like a fossil, that still have some chemical heterogeneities that are indicative of the initial heterogeneities that we know in living organisms. I'd like to show you one other illustration. This one will be much shorter, but just to illustrate how we can combine on different type of systems, on a different type of system, how we can combine transmission electron microscopy with X-ray microscopy to study minerals that are formed by a living organism. This is very new, actually. It's not been published yet. I mean, yeah, not even submitted, or maybe today. But so we have been looking at stromatolites from Lake Alchichica. This is in Mexico. This is a crater lake. And on the shore of this lake, we can find these stromatolites, so these really abundant formations that look like this, actually. They are carbonate deposits. They look green when you have a film of microbes populating the surface of these stromatolites. But when they die, the green is removed, the microbes fade away, and it looks white, like this. So all those carbonates we think are formed in association with microbes. We don't know how, but this crater lake is a pH 9, high magnesium. So the magnesium over calcium is 40, which is huge. And basically the mineralogy of these things is aragonite, so the calcium carbonate, plus a hydrated magnesium carbonate, hydromagnesite. So those things are a mixture of these carbonates. There is a huge microbial diversity in these samples. If you take a piece of these green rocks, if you look by a light microscopy at this green stuff, you have many things. I mean, it's really moving, living, with many, many microbes, different type of microbes, of filaments, spheres, and so on and so on. If you use epifluorescent microscopy, you can see those false colors, but showing that these are different pigments from different bacteria. So there are very many things. We have been doing some molecular analysis of the microbial diversity in these samples. You have thousands of different species. Now the question is, who in this big mass of microbes, or I would say a big mess of microbes, who is doing the calcification? Who is producing those stromatolites? I have no answer to this, okay? But that was the goal. That's what we were driven by. If you look by microscopy, look at just these samples by microscopy. This is light microscope. And we see these funny road-shaped microbes. By light microscopy, you can see that there are some dark granules within the cells. I guess most of microbiologists would say, okay, intracellular granules, we know that. This can be organic polymers, sulfur inclusions, and so on and so on. Now, okay, we are mineralogists, so let's have a closer look at this. This is SEM, and this is backscattered electron imaging. You see these road-shaped forms. They are here. And these are the granules. They are bright. If they are bright, that means that they are composed of heavy elements. Okay, so this is not organics, this is minerals. And what we could find, what we could say from here, and I'll show you that later, these are minerals. So those microbes, they form intracellular minerals. We could identify those microbes. They have pigments, they have chlorophylls, they are bacteria, they are cyanobacteria. So here we have cyanobacteria that form intracellular minerals. What are those minerals? We can first do transmission electron microscopy. So this is the Roche form. This time they are dark because this is transmission, so they are highly absorbing minerals, so they are dark, they look dark. We can do diffraction. Basically, we see nothing. This is amorphous. These are amorphous crystals. Now we can do EDX, map these things, and we see that those minerals, they contain calcium, strontium, and barium, and magnesium. So poorly crystallized contain calcium, barium, and strontium. Now, if we use STXM, X-ray microscopy, we can have a look at those samples at the carbon key-edge. And if we do the spectra, this is how it looks like. So you see a cell here, and in blue you have the inclusions, the mineral inclusions. In the yellow part, you have this spectrum. In the blue part, you have this spectrum. You can compare by yourself. The single difference between these two spectra is this peak here. This peak is at 290.3 electron volt. This is characteristic of carbonates. So those inclusions, they are carbonates. We know from TEM that they are calcium, magnesium, strontium, and barium carbonates. If we look at now we move, instead of looking at the carbon, we look at calcium by STXM, and we get spectra. This is the spectrum we get from the inclusion. This is how it looks like. Now we look at spectra from reference calcium, strontium, barium carbonate. Calcite, even if you don't know anything about this, they don't look the same. Aragonite, they don't look the same. Strontianite, they don't look the same. Benstonite, I don't know if you ever heard about that mineral. 6 months ago, I didn't know anything about that mineral. I don't know much more now because it's kind of unusual, very unusual mineral, actually. But this benstonite, the funny thing is, this is the composition of benstonite. Strontium, barium, magnesium, calcium, carbonate. Looks exactly the same spectrum at the calcium LH. So here we have cyanobacteria that form intracellular, amorphous, benstonite-like minerals. Why is that important? First of all, remember, in these stromatolites, those stromatolites I've seen you on the shore, they are composed of hydromagnesite and aragonite only, no benstonite. Within these cyanobacteria, you have benstonite. That means the chemistry is completely different within the cells and outside the cells. So here we have a new case of intracellular biomineralization by bacteria. This adds up to the very few examples we have, which are the magnetotoxic bacteria. Second thing, so far, cyanobacteria calcification. We know that cyanobacteria induce calcification, induce the precipitation of carbonate, of calcium carbonates. But so far, everybody considered that every time this calcification was extracellular. What is the implication of that? Then people have been looking for traces of extracellular calcification of cyanobacteria in rocks. So they were looking for fossils of cyanobacteria in rocks, thinking that they had to produce extracellular carbonate. So the way it will look like is that I will see a cyanobacteria cell surrounded by carbonate minerals. The problem is that when they have been looking for that, they have not found any cyanobacteria in the fossil record earlier than 700 million years old. We really believe that cyanobacteria are older than 2.5 billion years old because of the production of oxygen in the atmosphere. People have been looking for such cyanobacteria in the fossil records at 2.5, 3.5, they have seen nothing. The funny thing is that the cyanobacteria I've shown you, they are very deep branching in the tree of cyanobacteria. They appeared, and the ancestor was older than the chloroplast, for example. So they are close to they branch very close to the ancestor. The idea is that maybe the ancient cyanobacteria, they used to produce intracellular precipitate. And so there's no way you can find the ancestor of cyanobacteria as extracellularly calcified cells. So that's what I want to finish with. Tomorrow I'll show different type of story on iron oxidizing bacteria using STXM and TEM. And I'd like to finish with acknowledgements. I'd like to thank, of course, the European Association of Geochemistry and the Charles University for hosting me and organizing that seminar. Marie-Aude for all the work she did. Martine for hosting me and arranging everything. And all these co-workers that have been contributing most significantly to the work I've been showing you. Thank you very much.